Good morning, everyone. A very lovely good morning to all of you. Yeah, so is my voice audible to all of you and the screen visible to all of you? Yeah, good morning, Amit. How are you doing? Yeah, good morning, Dr. Sanjay. A very lovely good morning to all of you. So now let's quickly start up with the class. So, guys, yeah, good morning, Dilkush. How are you? Okay. So till now, guys, we have started up with the carbohydrate metabolism and we have covered almost 95% of carbohydrate metabolism. And now we are going to end the topic and we will be discussing the last topic on this. And yesterday, before winding up the class, I told you guys to just go through this because carbohydrate metabolism is very, very important from examination point of view. Okay, so today we are going to start up with the alternative pathways of oxidation of glucose. Alternative pathways. Okay, so before starting up with the alternative pathways, beta, let me just give you a brief thing. Ki there are major pathways. Major pathways means, yeah, good morning, Dr. Akhilesh. Major pathway, as it is clear from the word itself, what is it? It will be simple. All the molecules of glucose, yeah, whatever we are eating, having in our diet as carbohydrates, they will undergo the major pathways, which include glycolysis. So oxidation will be there, right, leading to ultimately TCA cycle. Then excess amount of glucose will be stored to form glycogen. And whenever energy is required, breakdown of glycogen will be there, which is glycogenolysis. Right? And then I told you one more process was there, gluconeogenesis, which basically occurs again during fasting when the body requires glucose. So these were the major pathways. Now, the minor or the alternative pathways will include HMP shunt, and uronic acid pathway. Right. And along with HMP shunt and uronic acid pathway, we are going to discuss today galactose and fructose metabolism. Right. I hope it is clear. Okay, so coming over to first of all, we are going to discuss HMP shunt. Now, since this is a minor pathway, or we can call it as an alternative pathway, that means that it is occurring in selective organs. The first and most important feature about this. Okay, and second is it is going to give rise to those intermediates which will be required by our body. Okay, so again, why I have written HMP shunt here? Otherwise, normally I write TCA cycle, I write glycolysis, shunt. So, what do you mean by the shunt? Now, beta, there are actually two shunts which are operative in the body. One is this HMP shunt, and the other one is RLS shunt. One student asked me about what is this RLS shunt also. Since we are discussing the shunts, I'll just tell you. RLS stands for, yeah, good morning, Mohammed Abdullah. Rapoport lubricant shunt. Shunt is that process in which there is no end product. Very important. Shunt does not have an end product. Yeah, good morning, Ritvik. How are you? I can see all the new joinees today. I think you people are attending the class for the first time, if I am not wrong. Dr. Akhilesh, yeah, Dr. Sanjay, good morning. Ritvik, right. So, shunt beta kya hoti hai? Shunt is the one which does not have an end product. That means obviously it is going to form certain intermediates which are required by the body, which are taken up by the body, and once the function is over, it's over. Right. So this HMP shunt is the shunt which does not have an end product. Same is the case with RLS. It is a shunt which does not have an end product. This occurs in RBCs, which we have clearly discussed. And this plays a very, very important role in hypoxia. Now coming over to, since the topic is HMP shunt today, what is it? Now this stands for Hexose Monophosphate shunt. And the other name of this is PPP, Pentose Phosphate Pathway. But this is the one which is the widely accepted terminology about this. Okay. Now coming over to this, the most important thing is it is a anabolic process. Right. And the most important thing is it is multi-cyclic in nature. It is a multi-cyclic process. Yeah, good morning, Vrishali. Yeah, good morning. Right, how are you? So it's a multi-cyclic process. But a multi-cyclic process kya hota hai? Which always has more than one substrate. So it always starts with 3 molecules of glucose, 6-phosphate. Clear? And then the substrate or the precursor is glucose, obviously. Clear? And glucose which gets converted to glucose 6-phosphate. That is what I've been telling you, that whenever glucose enters inside the body, since it is soluble in water, it always undergoes phosphorylation. And the process of attachment of phosphate is so that it cannot go back. Once we put a charge, it will be unable to go back. Okay? So, and then it has two phases. What are the phases? It has one oxidative phase and non-oxidative phase. It has two phases, oxidative phase and a non-oxidative phase. Clear? Oxidative means which will require oxygen and non-oxidative means which does not require oxygen. Clear? Then if I'll just show you the diagram and then we will discuss it in detail. Okay. So this is basically the HMP shunt. Now before taking up with this diagram beta, I will just explain to you and then we will go over this diagram. Clear? So if I talk about what happens actually, glucose 6-phosphate. Okay, since 3 molecules of glucose 6-phosphate are there, right? So what will happen is these 3 molecules of glucose 6-phosphate will first of all undergo oxidative phase. Okay, and it will give carbon dioxide here and they are going to generate NADPH. Right. So I can say that 3 molecules of glucose 6-phosphate will undergo decarboxylation here as well as they are going to give NADPH. And then since 3 are there, so they will be converted into 3 molecules of having 5 carbon atom each. Okay, so they will have 5 carbon atom, 5 carbon atom and 5 carbon atom. So here comes the oxidative phase. Right? And then after this oxidative phase, the non-oxidative phase will start. Non-oxidative means which will not require oxygen. So what happens here? See, 5 carbon atoms plus 5 carbon atoms. This is equivalent to how many carbon atoms? 10 carbon atoms. So this 10 carbon atoms will split up into 6 carbon atom and first of all it will split up into 7 carbon atom and 3 carbon atom. Right? Then 7 plus 3 again 10. So this will again combine and this will split up into 6 carbon atom and 4 carbon. Right. And now this 4 carbon atom will combine with this 5 carbon atom to split up into again a 6 carbon atom and a 5 plus 4. How many carbon atoms are there? 9 carbon atoms. So 5 plus 4 equals to 9. So it will be 6 plus 3. Okay. So I can say that, yeah. Hi, good morning, Alia. How are you? How many molecules they will split, madam? 1 or 2? But they are 3 carbon atoms, no? So 5 plus 5, 10. So this will be 1, 1. Obviously, then this will be again, this will split up. So overall, we are saying how many molecules started with 3? It will end up with 3. So 1 6 carbon atom, 1 6 carbon atom, 1 3 carbon atom. Right. So 6 carbon atom is what? It is fructose 6 phosphate. 6 carbon atom is again fructose 6 phosphate. And 3 carbon atom is your... Okay, Acha, you have joined e-Gurukul. Fine, fine. Yeah. Yeah, there are complete lectures and in detail also. Here also I'm teaching in detail. So whatever you are missing in that, you can take it from here or vice versa. Whatever is missing here, you can take it from that. And 3 carbon atom is glycerol aldehyde 3 phosphate. Right? Got my point? Ritvik, it is clear? Yeah, my pleasure. Ritvik, is it clear, beta? It starts with 3 because here you can see, no? 3 molecules I have already written. So then it will split up like this. Okay. So this is your oxidative phase and this is your non-oxidative phase. So always remember, Ritvik, is it clear? If yes, you can put a thumbs up. But to remember HMP Shunt, there are 3 to 4 important points. Okay? Yeah, okay, good. So I can say that it has an oxidative phase, it has a non-oxidative phase. Oxidative means it requires oxygen. Non-oxidative does not require oxygen, right? So I have just explained this is an oxidative phase. A detail is written here. If you want to go in detail, I will show you the detail also. You can see here, dekho, glucose 6 phosphate is forming NADPH. And it is forming, yahan pe it has shown with 1 molecule of glucose. Actually, it starts with 3 molecules of glucose. That is why it is a multicyclic process. 1 second, Mohammed, ma'am, the non-oxidative phase is this, beta. This is the oxidative phase. So you can say that ribulose 5 phosphate is there, right? And then what will happen is it will combine. I'll just show you these reactions also. Basically, I just want to give you the gist of this. So this ribulose 5-phosphate, ribose 5-phosphate, everything is synthesized here. Then they combine with one another, 5 plus 5, and they split up into 7 carbon atom and 3 carbon. Then 7 plus 3 will combine. It will split up into 6 and 4. Okay. And then this 4 will combine with another 5 carbon atom. Right. And it will give this. Again 5 plus 4, 9. So it will give 6 and 3. So this is your 6 and this is your. Clear? And that is what is represented here. So even if you draw the HMP shunt when it is asked in the exam, even if you draw like this, the way I have drawn, you will get marks. Non-oxidative phase, Amit, I'll just explain. Non-oxidative means no oxygen is required. So we have 5 carbon atom, we have 5 carbon atom, we have 5 carbon atom. Okay. And why it is? Because 3 molecules of glucose are there, they have their different fate. Now this 5 carbon atom will combine with another 5 carbon atom. So 5 plus 5 is how many carbon atoms? 10 carbon atoms. So 10 carbon atoms will split up to form 7 carbon atoms and 3 carbon atoms. Right. Now this 7 and 3 carbon atoms will combine again with one another. So 7 plus 3 is again 10. So this 10 carbon atom will now split up into 6 carbon atom and 4 carbon. Okay. तो ये जो 6 कार्बन का एटम है, this is our first product, which is fructose 6-phosphate. Now, on the other hand, this 4 carbon atom will combine with 5 carbon. So 5 plus 4 is 9. So 9 will split up into 6 and 3. So basically what we get is we get 1 molecule of fructose 6-phosphate, again 1 molecule of fructose 6-phosphate and glycerol aldehyde 3-phosphate. Okay. Now here it clearly explains why it is a shunt. Why it is a shunt? Because we know fructose 6-phosphate and this, they all will enter glycolysis. Isn't it? Excuse me. Right. Getting my point? Up the question which will actually strike your mind, in fact, which should strike your mind is that if it is ending into glycolysis, what is its use? Glycolysis, so already it is already taking place. No, glucose is coming, glucose is being oxidized. Yeah, glucose is being broken via glycolysis. What is the use of this HMP shunt? Okay, up use. Now again remember one thing, nothing is a waste in the body. Always remember whatever we eat, it has to be consumed inside the body. If something is important, it will be regained or reabsorbed back by the body and rest will be excreted out from the body. If there is a disturbance in the balance, it will lead to a disease, and that is what is we are sitting for, to detect that disease. Clear? And that is what you also are studying, to diagnose a disease. Up, coming over to HMP shunt, in normal way में it is forming 2 molecules of fructose 6-phosphate, 1 molecule of glycerol aldehyde, 3-phosphate, which is again entering glycolysis. So what is the use or what is the significance of HMP? Okay, up significance is where it takes place. First of all, you should know before going through the significance what is the site of HMP shunt. So if we talk about the site where it occurs, HMP shunt occurs in the liver, right? The most important role it plays in RBCs. Very, very important. In the red blood cells. Okay. And another important feature is it takes place in the adipose tissue. right? It takes place in the lactating mammary glands, the mother who's feeding the baby, in the mammary glands. Liver, because liver is, I told you, the kitchen of the body. RBCs, the most important role is basically played in the RBCs, adipose tissue. Even it plays a role in the testes also. Okay, because it is required for the synthesis of the hormones in the body. Clear? So this is the site. In the liver, in the RBCs, in the adipose tissue, lactating mammary glands. Where it does not occur? It does not occur in the skeletal muscle. It doesn't have any role to play, significant role in the skeletal muscle, and does not take an skin. Clear? Now coming over to this, what is its significance? So the significance of HMP shunt is it generates, very, very important. Those who are studying in first prof, beta, full-fledged question is asked on HMP shunt. Simple, explain HMP shunt in detail. What is the difference between HMP shunt and glycolysis? What is the significance of HMP shunt? And you will be surprised to know that the questions have been asked in the Viva also. Practicals ke baad there is Viva, you know. Viva maybe the same questions have been asked. So those students who are attending the lecture and they are in the first prof of MBBS, they have to study these whatever topics we are teaching to you in detail. And those who are preparing for FMG, there the question is asked, the HMP shunt produces what? So the options will be given to you. So if you know the basics, you will be able to solve the questions, as simple as that. Okay. So it generates NADPH and ribose sugar. Ab beta, one thing always remember, it is always NADPH. Most of the students get confused between NADPH and NADH. Okay, I will write it here also, and not NADH. Now, if we look at both NADPH and NADH, difference kya hai? Difference is only of the phosphate, but phosphate creates the major difference. Why? Because phosphate is having energy. So that energy is provided by this NADPH and ribose sugar. Clear? Now, if I talk about this is the function, it generates this and this. Okay. Now, what is the significance of NADPH? So, this NADPH, what it does is, it is required for fatty acid synthesis, right? Then it is required for cholesterol biosynthesis. We all know cholesterol, isn't it? कहते हैं ना, cholesterol is increasing. This person has increased cholesterol. So what all remedial measures we take? We have to cut short fat in the diet. So many things are there. So you will be, surprised to know that cholesterol is synthesized in the body also. So required for cholesterol synthesis. Okay. Then required for steroid hormone synthesis. These are the functions of NADPH, and along with it maintains the most important function. It maintains the integrity of the cell membrane of RBCs. Integrity means it keeps the cell membrane of RBCs intact. Maintains the integrity of cell membrane of RBCs. Okay, so this is the significance of NADPH, which is only produced by HMP Shunt. Clear? And second is beta ribose sugar. So if we talk about ribose sugar is required for the synthesis of the nitrogenous bases of DNA and RNA. This is again very important. Ribose is the precursor of the nitrogenous bases of DNA and RNA. Is it clear? So these are the two significant features of NADPH and ribose which are synthesized by HMP Shunt. Clear? Now, if you see the shunt, now you have to remember the enzymes also, obviously, because whenever we talk about biochemistry, biochemical reactions, they are not possible without the enzymes. So what are the main enzymes here? Now, if I talk about the enzymes, okay, here it is not visible. I will just write the name of the enzymes here. Oxidative phase mein beta, it always requires glucose 6-phosphate dehydrogenase. The spelling is glucose 6-phosphate dehydrogenase. Right? And here the enzyme is Transketolase. You have to remember the spellings of and the names of the enzymes. Transketolase. Here it is trans-aldolase. And here it is again trans-ketolase. Clear? So I can say that, beta dekho, thank you Amit for giving me the thumbs up. जिनने देना है thumbs up, those who have understood, they can give me the thumbs up. At least I desperately look for the thumbs up from the students, so that at least I will have an idea that you people are clear with the subject, clear with the topic. Thumbs up you can give, smileys you can give. जैसे होता है ना कि whenever a teacher gives you chocolate, how happy you are, how excited you are. Okay, same is the case with teachers. Teachers also expect some things, isn't it? So here I can simply say that, just summarizing it, how many transketolase beta मतलब trans is transfer. किसका transfer? Keto group. So there is a transfer of keto group. That is why 7 carbon atom keto group is there. It is, what is the name of this? This is pseudoheptulose. So always remember, whenever we write heptu, U, so that means it is coming from ketone. Fructose, Fructose. Fructose is there, sucrose is there, pseudoheptulose is there, erythrose is there, erythrulose, ribulose is there. They are all having ketone as the functional group. Okay? Trans-aldolase, transfer of aldehyde. Again, transketolase, transfer of ketone. Now, always remember one important thing, that this transketolase requires TPP as a coenzyme. Transketolase will always require TPP, which is thymine pyrophosphate, as a coenzyme. Is it clear? So I will here also I will write TPP as a coenzyme. क्लियर हो गया बेटा? So I will just write it here also. Here I have written NADPH. Transketolase, this is a very important feature. Transketolase requires TPP as a coenzyme. Can anybody tell me, beta, what is TPP? Yeah, thank you for this emoticon, Mohammed. Can anybody tell me, beta, TPP kya hai? From where this TPP is coming? It is vitamin B1. Okay, and thereby I can say that if vitamin B1 is deficient, transketolase might not function properly. That is what is the function of the coenzymes. If B1, if the coenzyme required, coenzyme का मतलब is co means working in cooperation with the enzyme. So if it is not working in a proper way, so that means this reaction will be hampered. This is what you have to understand. Clear? Right? Now, coming over to this significance also, I have told you what is the significance. It generates NADPH. This you have to mug up by heart, not NADH. You know, for FMG and NEET-PG, the question comes like that HMP shunt is responsible for the generation of. The options are NADH, NADPH, okay, NADH plus H positive. So always remember it will only generate NADPH. Right? Now coming over to the next topic, just now I told you what is the difference between HMP shunt and EMP, right? Okay, EMP is what? What is the difference between HMP shunt and EMP? You know what is EMP, beta? It is Embedden Meyerhoff Pranas pathway, or in Desi language we call it as glycolysis. Right? Now what is the difference between HMP and EMP? So if we talk about the differences, HMP is a shunt, right? And EMP is obviously a pathway, glycolysis. Second important point here is that no ATP is produced. Right? And if we talk about EMP, ATP is produced. We all know that it produces 7 ATP aerobically and it produces 2 ATP anaerobically. Right? Then HMP NADPH is produced. Right? EMP में no NADPH is produced. No NADPH. Right? And the last important point is CO2 is always produced. Right? And here no CO2 is produced. So these are the main differences between HMP and EMP. I hope it is clear to all of you, right? HMP shunt and Embedden Meyerhoff Pranas pathway. Clear हो गया बेटा? Is it clear? Now coming over after differences, Then we come over to the next important point. What role it plays in RBCs. This is also very, very important. What role it plays in RBCs? I told you that the function is it maintains the integrity of the cell membrane of RBCs. मतलब उसको intact रखता है. It keeps it the way it is so that the function of RBCs is easily carried out. ठीक है? So what role? Answer is that it maintains the integrity of the cell membrane of RBCs. How? The question is how it maintains. अब now see what happens here is there is one. Yeah, thank you so much for the thumbs up. Yeah. So the main thing is beta how it is maintaining. So always remember, there is one peptide जिसको हम बोलते हैं, we call it as glutathione. आजकल ग्लूटाथियोन के इंजेक्शंस भी आते हैं. If any girl is attending the class, you must be familiar with glutathione injections. On Insta, so many reels are also there in glutathione injections. Function पता है क्या है? Glutathione is working as an antioxidant. It is very good for the skin. उसको intradermal देते हैं and it gives very a good glow on your skin. Okay, so what is glutathione? It is basically a tripeptide, right? It is a tripeptide. Mane, it is formed of 3 amino acids. And what are those amino acids? Glycine, cysteine, and glutamate. Right. Up now see, we all know, देखो सब केमिस्ट्री पढ़ के आए हैं, you all know हमारी बॉडी में typical redox reactions आते हैं. Means if something is oxidized, the other is reduced. So this glutathione also exists in 2 forms. What are those 2 forms? एक होती है oxidized. So we write the oxidized form of glutathione as this. And reduced form of glutathione is this. Right. So this oxidized form can be converted to reduced form easily. Right. And what we require is we require one hydrogen here. From where hydrogen is coming? This hydrogen is coming from NADPH. That is why that pathway, NADPH is produced, not NADH. So this NADPH will simply give its hydrogen here. Like this. Okay. And from where this NADPH is coming? It is coming from HMP Shunt. And the name of the enzyme here is glucose 6-phosphate dehydrogenase. Okay. Up now, when glutathione is in its reduced form, ये क्या करेगा? It will simply, this glutathione will simply break this hydrogen peroxide. Okay, and this will break it into water and oxygen. Right? So I can say that all the processes interrelated. What is happening is NADPH, which is coming from HMP Shunt, it is converting reduced oxidized form of glutathione to reduced form. When glutathione is in its reduced form, then it is degrading H2O2 to water and oxygen. Clear? And if I have to write the name of the enzymes here, since reduction is occurring, so it is glutathione reductase. Right? And here it is glutathione peroxidase. Glutathione reductase requires vitamin B2 and glutathione peroxidase requires selenium as a cofactor. So I can say this is the reaction which is taking place on the cell membrane of RBCs. Now the question arises how it maintains the integrity. That is what I am telling you. It will maintain the integrity of the cell membrane. Now see, this is the normal reaction. Okay, if I talk about any abnormal reaction, if I say G6PDH is deficient, right? What is G6PDH? It is the enzyme which is generating enzyme producing NADPH. So if it is deficient, that means no NADPH is produced. Right? If I come here, that means this is not produced. If NADPH is not produced, this reaction will be blocked. Right? If this reaction is blocked, this reaction will also be blocked. It is all going stepwise. If I say G6PDH is absent, then what will happen? It will not generate NADPH. If NADPH is not generated, glutathione reductase will not work. If glutathione reductase does not work, glutathione peroxidase will not work. If I say this also doesn't work, what will happen? The concentration of this will increase, isn't it? Because if H2O2 is not being degraded into water and oxygen, its concentration will increase. Clear? And you know, when the concentration of, I'll just write no NADPH produced. So in a nutshell, increased concentration of hydrogen peroxide is there. और you know, ये जो hydrogen peroxide है ना, this is a very, I should say, important metabolite which is produced in the body. So if H2O2 is not degraded to water and oxygen, it gets converted to peroxide free radicals. Peroxide free radicals. You know what are free radicals? They are free. Free radicals always in search of an electron. Free radicals are always searching for an electron. ऐसे चलते हैं. So suppose this is the cell membrane of RBCs. So what will happen? They are searching electrons. यहाँ से they will get any electron. So what they will do is they will try to form a bond with it and breaking the cell membrane. Getting my point? I'll just repeat it. If free radicals are the ones which are having a free electron. So what is happening is whenever they are in search of an electron so that they can form a bond easily. So in bargain of searching an electron, they form a bond and they try to eat the cell membrane. If the cell membrane of RBCs is broken, it is eaten, it will lead to what? Hemolysis. Getting my point? So I can say that if whenever there is increase in the peroxide free radicals, it will lead to hemolysis. Lysis means splitting up of hemoglobin. So if hemolysis is there, automatically the count will decrease and it will lead to hemolytic anemia. So I can summarize it in a clear and simple language that deficiency of glucose 6-phosphate dehydrogenase will lead to hemolytic anemia. And this is very, very important. And it is a X-linked disorder. That means it occurs mainly in the male, because we know that if we talk about females are homogametic and males are heterogametic, isn't it? Homogametic identical gametes XX. Males में क्या होता है? XY is there. So this is X-linked, which is present in the males. So this is the most important point, that hemolytic anemia caused due to G6PD-H deficiency will lead to, but means hemolytic anemia will be caused due to the absence of G6PD-H deficiency. Getting my point? And that is why if I talk about here, if I take you back here, even B2 deficiency can lead to anemia. But main is this. Similarly, selenium also plays a very important role. How? By acting as a antioxidant. Okay, selenium is the trace mineral. Trace minerals वो होते हैं which are required by the body into small amounts. So even it is a trace mineral, it is required in smaller amounts, but still it plays a very important role in the body. And what is the role? It helps in working as an antioxidant. I hope it is clear to all of you. Or we also call it as, ये जो यहाँ पे है ना, deficiency of B6. So main reason is the oxidative stress. I won't go into that much detail, but this much you have to remember, beta, that deficiency of G6PD-H, which is coming from what? This is the enzyme, or what is its function? It is simply synthesizing what it is simply synthesizing. Can anybody tell me? Yes, anyone? Let me see how much you have understood. बेटा सिंपल कंसेप्ट है, देखो सिंपलेस्ट लैंग्वेज में, इफ देर इज absence या deficiency of G6PD-H, it will lead to no production of NADPH. NADPH will not be produced, then glutathione reductase will not act. If glutathione reductase will not act, glutathione peroxidase will not act. If glutathione peroxidase does not act, it lead to splitting of H2O2 into free radicals. The moment free radicals starts producing in the body, they will start degrading their cell membrane, and since they are maintaining the integrity of membrane of RBCs, so it will lead to hemolysis. It will lead to splitting of the RBCs membrane. Oh, thank you so much. Yes, and better keep on having this diet only, and you will see that even nowadays, so everybody is switching over to vegan diet. You see, the concept of vegan diet has come into function. People are leaving non-veg due to so many other reasons. And you will feel better, Vrishali. Okay, my pleasure, beta. Right? So, ye clear ho gaya? I hope this is clear to all of you. So, this is what is your HMP Shunt. Now we will just take 5 minutes break, and after break we will just conclude this HMP Shunt. Okay. So just take 5 minutes break and we will be back after that, guys. In the meantime, you can just relax yourself. Right. Yeah. So deficiency of G6-PDH will lead to hemolytic anemia. Okay. And it mainly occurs in the males, and main reason is oxidative stress. Now, beta, what is this oxidative stress? Simple in a layman's language, I would just like to tell you stress due to oxidation means related to free radical. Bas, you have to remember this. Slowly, you will learn this. Why? Because just now I told you कि due to hemolysis is there. अब देखो those again who are attending my lectures from day one, in glycolysis also I told you that deficiency of pyruvate kinase, leads to again hemolytic anemia. So what is the difference between these two type of anemias? Both are hemolytic, isn't it? Ab, what is hemolytic anemia? Hemolysis. वैसे तो there is so much to explain, but I'll just tell you in a simple language. Hemolysis is there, so it leads to decrease in the count, leading to anemia. But what is the difference between pyruvate kinase and G6-PDH? Clear? The main difference is no Heinz bodies are there. And in G6-PDH, Heinz bodies are present. The most important difference. Now, the question is, beta, what are these Heinz bodies? Okay, what are these Heinz bodies? They are basically, in the RBCs, this type of body starts occurring like this. So basically, these bodies are making this hemoglobin denatured. So the heme is denatured. Right? Denatured HP is there. Is it clear? Beta, is my voice audible to you, all of you? Because I can see here the screen is not visible. 1 second. Right? So whenever the heme is denatured, Heinz bodies are formed. So Heinz bodies are present in G6-PDH deficiency, but not in pyruvate kinase deficiency. Rest is the same. Hemolytic anemia is here also. Hemolytic anemia is here also. Right? And third important difference is that we can say that this is due to oxidative stress. This is due to oxidative stress, right? And pyruvate kinase due to pyruvate kinase deficiency. I hope this is clear to all of you. Any questions related to this? So I can simply say that that HMP shunt, which is an alternative pathway in the oxidation or for the oxidation of glucose, occurs mainly in the liver, in the adipose tissue, in the RBCs, right? And what is its significance? Its significance, it generates NADPH and it generates ribose sugar. Clear? Right? Any questions, beta? If there are any questions, you please ask me. Is my screen visible to all of you? Vrushali, is my screen visible, beta? Okay, right. Now, the another important feature of this is about transketolase. Now, I told you, beta, transketolase क्या है? Beta, please reply because is my sound audible? Can you hear me, guys? Okay, right. So then we come over to transketolase, right? So if we talk about this enzyme, I told you that TPP is the main coenzyme of this reaction. Clear? Now, since TPP is the main coenzyme, I can simply say that this plays a very, very important role in RBCs. Okay. So, if this B1 is absent, this is vitamin B1, no? So, if B1 is absent, so what will happen that this TPP will not work? And if TPP cannot work, what will happen? There will be a decreased activity of transketolase enzyme. Getting my point? So I can say that deficiency of TPP can be easily assessed by the decreased activity of transketolase enzyme in the RBCs. I hope it is clear to all of you. So this is all about HMP shunt. So if I have to summarize this HMP shunt, I can simply say that what are the main postulates of this? एक तो the main is it is a multicyclic process, right? Mainly it occurs in the liver and RBCs. Then what is the significance? It generates NADPH and ribose sugar. This is the main important point. Okay. And then another important point is that it has two transketolase reactions and one transaldolase reaction. Two transketolase reactions requires TPP, right? And then absence of G6PDH will lead to hemolytic anemia. And this hemolytic anemia is characterized by Heinz bodies. And I told you what are Heinz bodies. Heinz bodies are present, basically the denatured hemoglobin is there. Clear? Now I am going to ask you, what is the rate limiting enzyme here? Now you are going to tell me, beta, what is the rate limiting enzyme in HMP shunt? Can anybody tell me what is the rate limiting enzyme here? Yes, anyone? Vrishali, anyone? What is the rate limiting enzyme here, beta? No one? Arey beta, rate limiting enzyme, only one enzyme I am talking about. Glucose 6 phosphate dehydrogenase, isn't it? So the rate limiting enzyme of HMP shunt is G6PTH. So this is all about HMP shunt. I hope it is clear to all of you. And here, if we talk about the energetics, we can simply say it gives 6 carbon dioxide molecules and it gives 12 NADPH. That is all what you have to remember in this. Then after this comes uronic acid pathway. Right. Now, if we talk about uronic acid pathway, beta, again, this is an alternative method. So this is also a, chalo, thanks for giving me a thumbs up, Vrishali. It's an alternative method. This is very, very important again. Alternative method for glucose oxidation. Okay. Then second important point about this uronic acid pathway is ये होता क्यों है? Why it occurs and where it occurs? So mainly it occurs in the, if I talk about the site, it occurs in the liver mainly. And what is its significance? जैसे HMP shunt. The significance of HMP shunt is it generates NADPH and ribose. Here the significance is it produces glucouronic acid. It produces glucouronic acid. And what is the function of glucouronic acid? It helps in heme degradation, which we will be discussing. Heme degradation may it helps. And one more important point which you people studied, that it leads to the synthesis of mucopolysaccharides. So these are the two important features of glucouronic acid. And how glucouronic acid is produced? It is produced by uronic acid pathway in the body. If uronic acid pathway is there, then only glucouronic acid can be produced. And in non-primates, this pathway forms vitamin C. Right? In non-pathway, this pathway forms vitamin C, but vitamin C cannot be produced in human beings. This is also very important. Cannot be produced in humans. Why? Because we lack, means हमारे में ये एंजाइम प्रेजेंट नहीं होता है. Which enzyme? We lack L-glucoronidase. lactone oxidase, right? I'll just summarize it. What I'm trying to tell you here is there is one more alternative pathway of oxidation of glucose, which is uronic acid pathway. Now, this only takes place in the body just because of one simple reason, that it produces glucouronic acid in the body, right? And what is the significance of glucouronic acid? It helps in the degradation of heme and it leads to the synthesis of mucopolysaccharides. Clear? And in non-primates other than humans, this pathway forms vitamin C. We cannot form vitamin C. That is why we take vitamin C from outside. Why we cannot synthesize vitamin C? Because we lack this enzyme. Human beings lack L-gluconolactone oxidase. Clear? And this is, I'll just show you the picture of uronic acid pathway. This is your uronic acid pathway. This one. Okay. And here you can say that, I'll just zoom it 1 second. Here you can say that it starts from glucose. Obviously the precursor is glucose and it forms what? Here you can see that it forms glucouronic acid. That's it. This is its significance. So यहाँ पे आकर it produces. And then you can say that कि end में जाकर it will produce, here it is not given actually, it produces ascorbic acid or vitamin C. Here they have only given glucouronic acid, which is required. Clear? I hope it is clear to all of you. Generally in the exam, this question is asked only in one-liner form, that the significance of glucouronic acid is what? So what is its significance? It leads to the synthesis of glucouronic acid, uronic acid pathway. Okay, and you can say that this is how. Then another important thing here is, what is the clinical correlation of this reaction? Okay, clinical correlation is what? That if xylitol dehydrogenase is absent, if xylitol dehydrogenase is absent, okay, you can see that this is the enzyme. I'll just show the enzyme to you. Here, this is actually dehydrogenase. So if this enzyme is absent, there will be accumulation of this xylitol in urine. So you can say that increased concentration of xylitol, which is basically a 5 carbon atom sugar, then it will lead to essential pentosuria. Pentosuria is excretion. Urea is urine. Pentose is 5 carbon atom sugar. So when there is excretion of a 5 carbon sugar in urine, it leads to essential pentosuria. So this is all about uronic acid pathway. I hope it is clear to all of you, beta. Clear ho gaya? Then after uronic acid pathway, because main here is HMP shunt and little bit about uronic acid pathway is required. Then after this we come over to the last segment, galactose metabolism and fructose metabolism. Now first of all we are going to take the metabolism of galactose. Right. Now, if we talk about galactose, we all know it is the precursor of milk sugar. Galactose is required for the synthesis of lactose. So, it is the precursor of milk sugar, right? Then second important point about this is if we talk about the absorption, it is fastly absorbed than glucose, right? Then third important point about galactose is that it is phosphorylated. Now this is again very important. It is phosphorylated in the presence of enzyme galactokinase. Clear? Now what is the metabolism of galactose? Now see beta, this is our galactose. Just now I wrote it will undergo phosphorylation. So it forms in the presence of galactokinase. The other name of galactokinase is GALC. ATP is required for the reaction, obviously because phosphate is there, and it will form galactose 1-phosphate. Right? Now this galactose 1-phosphate will react in the presence of UDPG. Now, do you remember what was this UDPG? Anyone? बता सकते हो बेटा ये UDPG क्या था? Let me see. बताओ. Beta, uridine diphosphate glucose, isn't it? Yes, very good. Exactly. So now what will happen? This galactose 1-phosphate and this UDPG, they will interchange because galactose and glucose are linked as epimers. That is what I have told you in the class. So here, simply when interchange will occur, it will form UDP galactose and glucose 1-phosphate. And this glucose 1-phosphate will be then converted to glucose 6-phosphate and glucose will be produced. On the other hand, UDP glucose, it is required for the synthesis of other compounds. And if we talk about the enzyme here, enzyme is galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase, right? And this galactose on the other hand will be converted into galactitol. And the name of the enzyme here is aldol reductase. So this is the entire metabolism of galactose. First is galactose will be phosphorylated to form galactose 1-phosphate. The name of the enzyme is galactokinase or GALC. Then galactose 1-phosphate will combine with UDPG to form UDP galactose and glucose 1-phosphate. This glucose 1-phosphate will be converted to glucose 6-phosphate and producing glucose. On the other hand, UDP galactose will be required for the synthesis of lactose, other conjugates like you can say galacto-uronic acid is there. There are so many compounds which will be produced. It will be metabolized as such. And the short form of this is GAL. So one is your GALC, other is your GALT. Clear? So I can simply say that this is how galactose is metabolized. First of all, in the presence of aldol reductase, it forms galactitol and it forms galactose 1-phosphate will combine with UDPG to form UDP galactose and glucose 1-phosphate. Right? Now, this is the metabolism. Now, what is the clinical correlation of this? Very, very important. Clinical correlation. Now, if we talk about this, there are three enzymes which are responsible. So if I say that galactokinase is absent, or I can say galactose 1-phosphate uridyl transferase is absent. Either this enzyme can be absent or this enzyme can be absent. What will happen? It will lead to the increased concentration of galactose. It will lead to increased concentration of galactitol. Isn't it? So, whether galactose transferase is absent or galactokinase is absent, what will happen? Since the concentration of galactose will increase, we call it as galactosemia. Clear? So I will simply write that deficiency of galt, okay, or deficiency of gal, it will lead to galactosemia. Very, very important. अब now see beta, if we talk about this galactosemia, I can simply say, अब देखो, whenever we talk about any disease, we have to focus on the deficient enzyme first, and then we have to focus on the clinical features. So if I talk about the differences between the two, I can simply say that deficiency of GAL, ठीक है, GAL का I'll write the full form, galactose 1-phosphate uridyl transferase. It will lead to classic galactosemia. Okay, now coming over to the reactions here. अब now see if the concentration of galactose is increasing. Okay, सबसे पहले तो I can say this glucose will not be produced. But just try to understand how one thing is interrelated with the other. What I am trying to tell you, if this enzyme is absent, पहले चलो we'll take this enzyme. Okay, if galactose 1-phosphate uridyl transferase is absent, galactose 1-phosphate ka concentration will also increase. Okay, so this phosphate will not be available for other reactions. If this phosphate is not available, glucose will not be produced. The person will go under hypoglycemia. Ab, we all know that clinical features, okay, we all know that if lactose is not produced, for example, because this is milk sugar only, galactose is going to form lactose, so lactose won't be produced, so it won't be digested also, so it will lead to vomitings. Vomitings will be there. Then diarrhea will be there, right? Then you can say that jaundice will be there. Halke, this jaundice is different from the physiological jaundice. Okay, jaundice will be there. And the most important feature, again, the most important feature will be the concentration of galactitol increases. Because this is all interrelated. And always remember beta, whenever the concentration of galactitol increases, it leads to the formation of cataract. So here cataract will be there, which is also known as bilateral cataract. Clear? So this is classic galactosemia, where the deficient enzyme is galactose 1-phosphate uridyl transferase. Okay, so there is increased concentration of galactose leading to hypoglycemia, vomiting, diarrhea, jaundice, and cataract. Breastfeeding is contraindicated in this. Okay, and then the most important is cataract. Second, we are talking about the other one. If I say this is absent, galactokinase is absent. So if I talk about only this enzyme, The increased concentration of galactose will be there, right? And the formation of cataract will be there. So if I have to differentiate between the, you can say deficiency of GALC and GALT, GALC may only cataract will be visible. Only cataract is visible. Right? So this is the metabolism of galactose. And if we have to go for laboratory investigations, what are the laboratory investigations? The laboratory investigations are, the most important one here is beta, Benedict's test will be positive. Benedict's test will be positive. Clear? Then if Benedict's test is positive, which is for reducing sugar, we might get confused between galactosemia and diabetes. For that, we carry out glucose oxidase test, which is only for glucose. And glucose oxidase test will be negative. It has to be negative because it is not a case of diabetes, it is a case of galactosemia. Okay, and then mucic acid test. Mucic acid test, which is specific for galactose, it will also be positive. And then we can carry out the enzyme studies. Clear? So this is the metabolism of galactose. I hope it is clear to all of you. Right? Then after this, we come over to the last topic of this. You can say fructose metabolism or metabolism of fructose. Now, if we talk about fructose metabolism, the most important feature here which we have to remember is that it is the most lipogenic sugar. Very important. Second important point is it is more sweeter than glucose. More sweeter than glucose. Right? Then again, if we talk about the metabolism, simple is fructose will undergo phosphorylation first of all. And it will be phosphorylated to form fructose 1-phosphate. And the enzyme is fructokinase, where ATP is converted to ADP. Right? And this fructose 1-phosphate will split up. Right? The name of the enzyme here is aldolase B. And fructose 1-phosphate splits up into glycerollaldehyde and dihydroxyacetone phosphate. Obviously, both are the intermediates of glycolysis, so they will end into glycolysis. Clear? So I can say that fructose, the moment it enters, because whatever syrups, sweet syrups, जैसे हम लोग cough syrups and all, which are sweeter to taste, they are all rich in fructose. So fructose also has to be metabolized. So this fructose will be phosphorylated in the presence of fructokinase to form fructose 1-phosphate, which will split up into glycerollaldehyde and dihydroxyacetone phosphate, which will merge into glycolysis. Clear? This is how it will be metabolized. Now, after this, what will be the clinical correlation of this? the clinical correlation is either this enzyme is absent or this enzyme is absent, isn't it? If this enzyme is absent, this concentration will increase. If this enzyme is absent, this concentration will increase. Clear? So if I have to write that in fructokinase deficiency, what will happen? Essential fructosuria will be caused. Essential fructosuria is caused means there is excretion of fructose in urine. Clear? Essential fructosuria is excretion of fructose in urine. And if we talk about the second enzyme in aldolase B deficiency, in aldolase B deficiency, hereditary fructose intolerance is caused. Hereditary fructose intolerance. Okay, we designate it as HFI. So we can say that HFI is caused due to absence of aldolase B. Now, this is very important. What are the clinical features of HFI? Okay, when the child is born with the deficiency of HFI, no symptoms are visible. So, initially, the patient is asymptomatic, means no symptoms he will show. Is asymptomatic. But when fruit juice is given, when अब देखो initially he won't show any symptoms क्योंकि normally glucose and all are being metabolized, but when fruit juice is given, his condition deteriorates. His condition deteriorates. Becomes worse. Why it becomes worse? Because he cannot digest fruit juice. He cannot digest sweet things. And why he cannot digest? Because the enzyme required to degrade it is absent. Getting my point? So I can say that in aldolase B, hereditary fructose intolerance is caused. Initially, the patient does not show any symptoms, but when fruit juice is given, his condition deteriorates. Okay, and it leads to what? Basically, it leads to what are the clinical features? Again, same. Vomitings will be there. Flatulence will be there. Diarrhea will be there. Okay, almost all symptoms will be similar to that of galactosemia, but no cataract is there. Is clear? No formation of cataract will be there, and obviously hypoglycemia also will be. This is hereditary fructose intolerance. And if we carry out what is the laboratory diagnosis? So if we talk about laboratory findings, right here, Benedict's test will be positive. Yes or no? It will be positive. Why it will be positive? Because fructose is also a reducing sugar. Then if we talk about glucose oxidase test will be negative because it is not a case of diabetes mellitus. And Selvanov's test will be positive. right? So these are the main tests which are carried out to check whether the person is suffering from hereditary fructose intolerance or not. Clear? So this is all about the metabolism of galactose, fructose and the alternative methods. So here we finish first segment of our biochemistry, metabolism of carbohydrates. I hope it is clear to all of you. Any questions related to that? If it is clear, then we will wind up the session here, and tomorrow we are going to discuss regulation of blood glucose. Right? So if there is any question, beta, you can ask me. If there is no question, then I will definitely wind up the session for today. Everything is crystal clear to all of you? Chalo, good to hear. Yeah. So thank you so much. So tomorrow also we are going to meet at the same time, and tomorrow I will be taking up the regulation of blood glucose. Have a good day.